So, um, there is this very comforting lie that we all learned in high school biology.
Oh, just one.
Well, yeah, fair point. But I'm thinking of a very specific one. It's one of those things that makes the world feel really organized and, I don't know, binary. On one side of the line, you have life,
Right.
which is, you, me, the bacteria in your yogurt, the tree outside. We eat, we grow. And crucially, we actually have the machinery to build our own bodies.
And then on the other side of that very neat little line, you have viruses.
Exactly. The obligate parasites.
Mm-hmm.
The genetic ghosts, I like to call them. Because the way I always learned it was that a virus is basically just a code in a box.
Mm.
It has the blueprints, sure,
Mm-hmm.
but it doesn't have a construction crew. It doesn't have a kitchen. If it wants to, say, cook dinner, it has to break into your house and use your stove.
Yeah, and that is the classic dogma. It's the definition that keeps virologists sleeping soundly at night, knowing exactly what a virus is and isn't. But today, for this deep dive, we are going to ruin that sleep. Because we're looking at a paper that suggests some viruses aren't just breaking into the house. They are actually bringing their own industrial appliances with them.
We are diving into a really fascinating study published just this year, 2026, in the journal Cell.
Yeah.
It comes from Fels and colleagues, and the title is, it's a bit of a mouthful, but it perfectly sets the stage. It's called Giant DNA Viruses Encode a Hallmark Translation Initiation Complex of Eukaryotic Life.
Which is, a very polite, highly scientific way of saying, hey, we found a virus that packs its own kitchen.
Exactly. So our mission for this deep dive is to unpack this discovery for you. We are going to look at a specific giant virus called mimivirus.
Such a great name.
It really is. And we're going to see how it breaks the fundamental rules of biology by carrying its own protein synthesis machinery.
We will also look at the secret handshake, basically a molecular password the virus uses to ensure only its own orders get cooked in that kitchen.
And finally, we'll talk about why. Like, why would a virus bother carrying all this heavy genetic luggage? And it turns out it makes them absolute tanks in hostile environments.
Oh, completely.
But before we get into the molecular spycraft, let's introduce the monster here.
Yeah.
Because we aren't talking about the common flu.
No, not at all. We are talking about Acanthamoeba polyphaga mimivirus, or APMV for short. And when we say giant virus, that is not hyperbole.
Just how giant are we talking here?
Well, normally you need an electron microscope to see a virus, right? They're incredibly tiny.
Right.
But mimivirus, you can actually see it under a standard light microscope. In fact, for years, scientists didn't even know it was a virus.
Wow.
Yeah, they looked at it and assumed it was a bacterium because of its sheer physical size and the massive size of its genome.
It's basically the Godzilla of the viral world.
Essentially, yes. And it hunts amoebae. These are single-celled organisms that live in some pretty rough neighborhoods. Think dirt, sewage, cooling towers.
Okay, so we had this giant virus infecting an amoeba. Now, usually when a normal virus infects a cell, the process is, well, it enters the cell, hijacks the ribosome, which is the cell's protein printer, and forces the host's workers to print viral proteins.
Correct. The virus provides the mRNA, which is the instruction tape, but the host provides the machine and the specialized workers to actually read that tape.
Okay.
And these workers are called translation initiation factors.
So think of these factors as the start button on the printer.
That is a perfect analogy. In eukaryotes, which includes the amoeba and also us humans, this team of proteins is called the EIF4F complex.
EIF4F.
Got it.
It's a group of proteins that grabs the instruction tape, unwinds it, and loads it onto the ribosome. Without EIF4F, the ribosome just sits idle. Nothing gets built.
So Fels and his team, the authors of this Cell paper, they wanted to see this hijacking in action.
Yeah.
They decided to peek inside an infected amoeba using mass spectrometry to analyze the ribosome, so basically looking at the factory floor during an active infection. And they fully expected to see the host's EIF4F complex doing all the heavy lifting.
Because that would be standard operating procedure.
Exactly. The virus gives the orders, the host does the work, but that is not what they found.
This is the real aha moment in the paper.
It really is. Alongside the host's machinery, they found 3 proteins sticking to the ribosome that simply did not belong to the amoeba. They were viral proteins.
So the virus didn't just bring the blueprints. It brought its own foreman and its own construction crew.
Precisely. And they dubbed this viral crew VIF4F. The V stands for viral, naturally.
Makes sense.
And it consisted of 3 key players that directly mimic the host's machinery. First, they found a viral helicase called VIF4A. That is the worker that unwinds the genetic tape so it can be read.
Good unwinder.
Second, a viral cap binder, VIF4E. That's the one who grabs the end of the tape to start the whole process. Right. And third, a viral scaffold protein called VIF4G that physically holds the whole team together.
I want to pause on that third one for a second, the scaffold, because the paper mentioned something really interesting about its shape.
Oh, the structural biology of VIF4G is fascinating. The researchers used AlphaFold, which you probably know is the AI tool for predicting protein structures.
Right.
And they also did actual physical crystallography. They found that the viral scaffold looks eerily similar to the middle domain of the human or amoeba version. It has these tandem helical repeats.
So if you picture it, it's like a curled ribbon structure.
Yes, exactly. It acts as the backbone that holds the helicase and the cap binder in place. But here's a kicker.
Yeah.
If you look at the actual genetic letters, the DNA sequence that codes for it, it is totally different from the host sequence.
So it's a case where the 3D structure is conserved because the shape works, but the underlying genetic language is completely divergent.
Highly divergent. And that tells us something huge. It means the virus didn't just steal this gene yesterday from a host.
Right.
It has been refining this tool for a very, very long time. It is not a cheap knockoff. It is a bespoke piece of molecular engineering.
So mimivirus has this custom-built machine, this VIF4F complex. But the obvious question that comes up is why?
Yeah.
Why carry all that extra genetic baggage if you are already inside a cell that has the exact tools you need? It just seems incredibly inefficient.
And evolution is usually ruthless about efficiency. You don't pack a tent if you're staying in a hotel.
Exactly.
Unless you know the hotel might kick you out.
Or unless you want to do some massive renovations that the hotel management definitely wouldn't approve of.
Spot on. The researchers found that the virus uses this machinery in a very specific, highly regulated way. It actually relies on a temporal switch.
A timing mechanism.
Yes. To figure this out, they used a technique called ribosome profiling. Think of it like taking a high-resolution snapshot of every single instruction tape being read by the factory at different times during the infection.
Like checking the factory logs at different shifts.
Exactly. And in the first 4 hours of infection, which we call the early phase, the virus is actually being quite lazy.
Lazy how?
It uses the host's EIF4F complex to translate its early genes.
So at the start, it is acting exactly like a normal virus, just saying, hey, can I borrow your stove to make a quick snack?
Right. It saves energy. It lets the host do the initial setup work. But then right around the 8-hour mark, the late stage, there is a hard pivot.
Wow.
The host's machinery gets completely pushed aside, and the viral VIF4F complex takes over the entire production line.
Why the switch at exactly 8 hours? What is changing in the infection?
8 hours is construction time. That is when the virus needs to start building the capsid, the hard outer shell, and the core proteins. Basically, the physical structural components of the new baby virus particles.
I see. So the host machinery is good enough for the software, the early regulatory genes, but when it comes to the hardware, the actual heavy lifting of building the bodies, the virus absolutely wants its own crew in charge.
It seems so. And to prove that this viral kitchen is actually necessary and not just a weird quirk, the researchers did something pretty ruthless. They performed knockout experiments.
This is where they go into the virus's DNA and intentionally delete the genes for the VIF4F complex.
Right. They deleted the genes for the viral helicase, the cap binder, or the scaffold. They basically broke the viral kitchen to see what would happen.
And what happened?
It was catastrophic for the virus. It could still replicate its DNA perfectly fine. It had the blueprints, but it couldn't build the house.
There is this image in the paper from the electron microscope that really stuck with me. Usually a viral factory inside a cell looks like a beehive.
Yeah.
It's just full of these hexagonal geometric shapes, which are the new viruses being assembled.
Right, those are the viral progeny.
But in the knockout version, the factory zone was there, but it was just empty.
Empty promises, no capsids, no shells, just a cleared out factory floor with absolutely zero product. It definitively proves that this VIF4F complex isn't a luxury item. It is essential for building the physical body of the virus in that late stage.
Okay, so we know what it is. It's a custom translation crew. And we know when it works, which is the late stage. But this next part is the part that really blew my mind, the how.
The specificity.
Right, because inside this infected cell, you have a massive soup of RNA. You have the host's mRNA floating around, and the virus's mRNA floating around right next to it.
Yep.
And both of them have a cap at the start, this chemical flag that says Translate me.
It's called an M7G cap. It is the universal standard start signal in eukaryotic life.
So if the viral machine, the VIF4F, just grabbed anything with a cap, it would accidentally grab the host's instructions too.
Exactly.
It would waste all its energy building amoeba proteins instead of virus parts. So how does it know which ones actually belong to the virus?
It uses a secret handshake, a molecular password.
I love this part. Please break down the password for us.
So the researchers looked really closely at the sequence of the viral mRNA itself. Almost all of the mimivirus late genes start with the standard cap, but immediately after that cap, at position +1, there is an adenosine, an A.
Just an A. That seems pretty common. Not much of a password.
But this A is chemically modified. It is 2-O-methylated.
Okay, we are getting into the biochemistry weeds here. 2-O-methylated. What does that actually mean for the listener trying to picture this?
Think of it like a standard house key, but with an extra little bump of metal welded onto the side. It's a specific chemical tag, a methylation mark.
So the viral instructions have this special bump on the key.
Yes. And the viral cap-binding protein, VIF4E, is designed perfectly to fit that exact bump.
How so?
When they analyzed the crystal structure, the 3D shape of the protein, they found that the binding pocket is tilted.
Tilted.
It's angled differently than the human or amoeba version of the protein. And inside that pocket, there are 3 specific amino acid residues: serine 213, methionine 215, and tyrosine 182.
You definitely don't need to memorize those names, but these 3 act basically like the tumblers in a lock.
Exactly. They form a very precise slot that perfectly accommodates that one methylated adenosine.
Whereas the host version of the protein just doesn't care.
The host version is promiscuous. It's an open door. It binds anything with a basic cap, but the viral version is a bouncer with a clipboard.
I like that.
It checks for the cap, and it checks for the methylation bump. If you don't have the bump on the key, you don't get in.
That is incredible. It is a brilliant mechanism of self-recognition. Just saying, only build my stuff.
It allows the virus to completely monopolize the resources. Think about it. Late in the infection, the cell is dying, energy is extremely low. The virus ensures that every single ounce of remaining energy goes into building viral capsids, not repairing the host cell.
Which brings us to the ultimate question of why. Because nature doesn't just evolve complex locks and keys for fun.
No, it doesn't.
It is energetically expensive to carry these extra genes around. There has to be a massive survival advantage.
And the advantage is pure resilience. We really have to remember where these amoebas live.
Right, the rough neighborhoods. Siberian permafrost, scalding hot springs.
Exactly. If an amoeba is floating in a hot spring, or gets hit with environmental toxins, or just starts starving, it triggers a severe stress response.
It goes into lockdown mode.
Exactly. The cell tries to save energy to survive the crisis, and the very first thing it does is shut down protein synthesis. It turns off its own EIF4F complex. Kitchen's closed, everyone go home.
Which, for a normal virus, relying on that kitchen, is an absolute death sentence. If the host shuts the kitchen down, the virus starves. It can't replicate at all.
But mimivirus brought its own kitchen.
So it just ignores the lockdown entirely.
Completely overrides it. The researchers tested this brutally in the lab. They literally starved the amoeba. They hit them with chemicals like saxagargin to cause ER stress, basically forcing the cell into a chemical panic attack. A normal cell would start making proteins immediately.
And a virus, how did it react?
The wild-type mimivirus just kept pumping out viral particles like nothing was happening. It did not care at all that the host was in full crisis mode.
That is terrifyingly efficient. It completely decouples the virus's survival from the host's health.
It does. And the definitive proof that the viral kitchen was the key to the survival came from the mutants. Remember those knockout viruses we talked about earlier?
The ones missing the VIF4F genes. With the empty factories.
Right. When they put those knockout viruses under the exact same stress conditions, they failed. Production dropped by a factor of 10 to 100.
Because they had to rely on the host again.
Exactly. They became dependent on the host. Because they lost their independent machinery, they were suddenly at the mercy of the host's stress response, shutting everything down.
The researchers even compared it to a cousin virus to prove the point, right? Pneumovirus.
Yes. Pneumovirus is another giant virus, very similar to mimivirus in a lot of ways, but it naturally lacks this VIF4F machinery. It's kind of like the control group provided by nature.
And what happened with it?
Sure enough, under stress, pneumovirus struggles immensely. It essentially stops replicating when the host gets stressed. Mimivirus, on the other hand, just thrives.
So this VIF4F complex isn't just a biological quirk. It is a hardcore adaptation for extreme environments. It allows the virus to turn the host into a zombie factory. The host might technically be trying to die or shut down to save itself, but the virus just keeps the machinery running.
It's like saying, I don't care if the ship is sinking, I am still finishing my dinner.
That is a very powerful image. And it really forces us to circle back to the intro. We started this deep dive with the definition of a virus as this completely helpless parasite.
And yet here we have a virus that has its own translation system. It has a secret code for recognizing its own genes. It actively functions and builds itself when the cell is basically dead in the water.
It feels like mimivirus is sitting in this really uncomfortable gray area. It's not quite a cell, but it is definitely way more than a genetic ghost.
It definitely is. The authors of the paper actually emphasize that this machinery gives the virus replication plasticity. It acts a lot less like a passive packet of data and much more like a competitive organism.
Like really blurring the lines.
It really is. The paper calls it an evolutionary innovation. The virus didn't just steal these genes recently and keep them frozen in time.
Right, the divergent sequence.
Exactly. It evolved them. It created that tilted pocket in VIF4E. It meticulously tuned the system for methylation recognition. This is highly sophisticated molecular engineering happening purely inside a virus.
So here is a final provocative thought for you and for you listening at home. If a virus can encode the ability to make its own proteins, at least the initiation part of it, what is the actual limit?
That is the billion-dollar question in biology right now.
Because if mimivirus has its own DNA repair mechanisms, which we know it does, and its own metabolism genes, and now its own protein synthesis starter kit, could it theoretically evolve to become a fully independent cell one day?
Or are we actually looking at the exact reverse?
The reverse.
Here? Yeah. Some scientists argue that giant viruses are actually the remnants of an ancient fourth domain of life that slowly lost its independence, that they used to be full cells and they are slowly devolving into viruses.
Oh, wow. So instead of a virus learning to walk, it's a runner deciding to just sit down forever.
Exactly. It outsourced the hard parts. But if it gained just a few more pieces back, maybe a way to generate its own ATP for energy, a full ribosome of its own, it simply wouldn't fit our definition of a virus anymore. It would be a totally new kind of parasitic life form.
A spectrum, not a binary.
It's a spectrum. Life is never as neat as the textbooks want it to be.
Well, next time you get a cold, just be incredibly thankful it is likely a tiny rhinovirus and not one of these microscopic giants bringing its own industrial forge into your cells.
Although, thankfully, for now, they really only infect amoebae.
For now. I love the optimism.
We do what we can.
On that note, thanks for joining us on this deep dive into the microscopic giants living in the dirt. It really does make you rethink what alive actually means.
Always a pleasure to explore the bleeding edge of biology.
Catch you on the next deep dive.